I think you're right. We're moving along those pathways, but sometimes we've taken choices a bit differently. And I want to tell our story a bit, show you some examples, but mainly try to bring over the message which choices we took and why we took them. Because I think we took some interesting choices, which might be interesting for some of the people in the room. So where do we start? We start back in 2005 with Wilfried de Bakker, who's the head of respiratory medicine at the University Hospital in Antwerp, who's also at EMA looking at some registration files, and in Belgium he's also judging some reimbursement of products. And he runs into a problem. He sees all these files having FEV1 as endpoint in clinical trials. And FEV1, you blow in a tube as hard as you can for 1 second, and that tells you everything about your lungs. And FEV1, not saying it's a bad measure, it's a very good measure for certain conditions, but there is limitations to FEV1. And I think in 2005, probably the sector was running into some issues, trying to register anti-inflammatory, anti-fibrotic drugs, bringing new bronchodilators to market which had to be compared to the old ones, and noticing that FEV1 might not be the optimal situation. He was talking at that moment to his son, Jan de Bakker, our CEO, study mate of myself, who was studying aerospace engineering, specializing in aerodynamics and flow simulations, working on solar cars, sailing boats, fighter jets, Formula 1 cars. And he was thinking, like, all this visualization of flow, breathing is flow. Can't we bring the two together? And that's where the idea started. At about the same time, I ran into Jan, and he told me the story. And I said, it's interesting. I was in Belfast doing an internship. And in Belfast, there was a project at that time running with Westpac around nasal flow simulations. So I told him about this. He said, actually, we have some funding at that time from Novartis for 2 PhD students for 2 years. He said, do you want to join? And that's where it started. 2 minutes, everything arranged, and off we went. I was still young, a bit lighter than I'm now. An office we didn't have. We were working from Jan's house in the hallway. Our lunch break, we played golf in the garden, just some fun. And we thought we were good at making presentations. When we look back at them, they look a bit outdated, but we were proud of doing something. And we had a goal. We wanted to develop a new biomarker for lung diseases, and preferably better, combining what we knew with what was going on in the field. And this is an important one. Our goal was really to come up with a biomarker. And in our belief, the definition of a biomarker, it's a measure that you can take from a patient, or something you can derive from a measure taken at a patient, an image, which tells you something about the disease state of the patient. That was the goal, very clear. Then we ran into some lessons. So the first lesson, go further than where other people go. First choice we had to make, which imaging modality? Because if you want to simulate flow, you need to have a model of a patient-specific airway tree. Well, very rapidly we ended up with CT, just because of the high resolution, the fact that air is a natural contrast in CT, and you can extract airway trees by segmenting them down from a CT scan. But back in 2005, that was difficult. It was a manual task, coloring the airways, going pixel by pixel, indicating that they were part of the airways. And if you zoom in, see a bit of the challenge. So you see the airways here, but you notice there's leakage into the parenchyma. So automating this, not easy. We didn't have an experience in image processing, but we wanted to do this. So we spent a lot of time in generating these models. We also realized quite quickly that CT scanner manufacturers at that time, they had some visualizations of the 3D structures, but the visualization is not the same as a real three-dimensional structure. Also, when you collect these voxels, you have to smooth them because running airflow simulations through those, that didn't work. So you needed to come up with something to make them more smooth. And in the end, generating one airway tree, starting from a CT scan, took us 1 week. 1 week of manual labor. Yeah, at that time point, we never thought about it. We said, this is gonna work. That was our line of thinking. A lot of people would have said 1 week per patient. It's impossible, because that was just to get the model. If you want to go further, well, you have to do the CFD run. Well, that took us a day of manual labor to set it up and to run it, and then you had the post-processing. Well, at least a week of manual labor to do it. But we were very happy. We had something that we thought could have value. But we wanted to stay focused, because at this point we could have made some choices. We could have said, let's spend time on image processing. Let's start implementing it. Let's spend time on automating the CFD code, make it better. But that was not the goal. The goal was to develop a biomarker. So these timing issues, we took them. Let's work with these and let's focus on what we want to do. We want to go for a biomarker. And what we thought, what could be important at that time is because we want to look at functionality. One scan was definitely not going to be enough. We wanted something of the dynamics of the airways. So we added a second scan into our protocols. We wanted inspiration and expiration, so we had an idea of the dynamics. Also, and this is a very important one, from the beginning we say it's about the patient. We're not going to assume anything. We're going to measure things. And if we cannot measure it, we work with what we can measure, and we're not going to assume things. This is work that we did together with University of Oxford, with Rafael Bordas, on adding one-dimensional networks, branching networks, to a segmented airway tree. Statistically, this is correct for a healthy subject. But you don't know if for this subject it adds value or it brings you further away from the truth. So we never did this. Never. Well, we've done this in research settings, but for the work that we wanted to do, we didn't want to assume things, we wanted to measure things. So we said, what can we measure? Well, based on the fact that we had expiratory and inspiratory CT scans, we could measure the airway tree, we could measure at the mouth how the patient was breathing, and in the lungs we knew how much % of the air was going to the lobes. Why the lobes are not smaller? Because a lobe you can identify with 100% certainty from the CT scan. You see the fissure lines. A segment, you start assuming things. You co-register expiration to inspiration, you start assuming things. We never did that. We stayed with the patient because we said it's patient-specific data and we want to know whether it correlates with the state of the patient. That was very, very clear choices for us. Also, we diversified. Anything we could lay our hands on, we would play with it. So we looked at sleep apnea. What's the airflow doing in the upper respiratory tract? We look at airways, pre-bronchodilation, post-bronchodilation. We did deposition work, virtual deposition work, correlating it with work that came from scintigraphy. This is work from Omar Usmani, which was published in 2015. So we tried to recreate that based on the patient data that we had. We saw that patients were using masks for breathing. What's the influence of a mask? Well, we tried to simulate airflow in that. So we tried a lot of things just to try and understand what our technology was doing. Also, from a business perspective, we diversified. If we saw an opportunity, we took it. First person we hired, we were 2 and we had work for 3. So we opened a position. There was an Indian guy who was in Virginia who said, like, I want to come work with you. He came. 3 months later, we had an office in India. Just try it. Nothing to lose. A couple of years later, we said, we have a lot of clients in the US. We opened an office in the US. Try to diversify. Try to take opportunities. And our presentations got a bit better. They started looking nicer. Then we had some interesting milestones. The first one was the first client. First study that we did, paid, was with UCB in Belgium, actually with the Chiesi compound carmoterol, where we looked at 10 subjects that were actively treated and 4 controls with an acute bronchodilator, and we looked at what's the effect of the bronchodilation and what's the stability if you don't do anything. The scans were taken at total lung capacity, so we wanted to look at the changes in structure and function for those airways. These were still the days that one scan took us 2 weeks to analyze. This was a huge amount of work, but we did it, and the results were massive, because everybody said looking at airway volumes that you can see from a CT scanner is irrelevant when you look at a bronchodilator. Bronchodilation happens much deeper down into the lung. That's what everybody said. But when we looked at what happened to the volumes, well, we saw that the volume went up quite a bit, and if you looked peripherally, so anything from third generation onwards, we saw massive responses. We saw bronchodilation on average 30, 50, 40. We saw a lot of things which nobody was expecting. With the increase in volume, we saw a drop in the resistance, which was interesting. Here are some examples from what we did back then. So you have the airway trees chopped up into pieces, and for every piece you see the change in volume from pre to post bronchodilation. So this is, well, let's start here. This is one of the patients. You see bronchodilation mainly in the more distal regions, and you see a response in FEV1. This is another patient treated. FEV1 108 already at baseline, increased a little bit, but still you see quite significant dilation of the airways, which was interesting for us because we didn't expect that. The field didn't expect that you would see bronchodilation at a point where FEV1 was not changing anymore. Control patient, also very interesting for us. FEV1 stable, airways stable, nothing ongoing. This was something that when we saw these results, we were talking to each other. We hoped that it would be good, but we never expected that it would be this good. That was the first big milestone for us. Second milestone, I think it's also mentioned before, try to publish our data. So our first publication was a big milestone. This was something we did around sleep apnea, but it was a big milestone. The next one, a returning client. We did some work with GSK on animals, first on rats and guinea pigs, and then they came back for work on monkeys. For us, that meant that we were selling something to the client that was of interest for them because they were coming back to us. Also an important point, validation. Also mentioned before by Sarah, validate what you have. So our approach of extracting airway trees from a CT scan, having dynamics from the inspiration, expiration, measuring the breathing profile at the mouth and releasing particles into the lungs is totally different than what you do with SPECT. But it measures the same thing. If you release the same kind of particles, it measures the same thing. And we saw that on a lobar level, we were less than 2% off of the SPECT measurements. This was for us very important milestone to say that one approach which measures the same but is coming from a totally different source is actually showing you the same as with the clinical measurement. Our presentations still got better, starting to be more professional. This one was for us also very interesting, because at that time people were saying, yeah, you can measure bronchodilation. Interesting. But an anti-inflammatory compound, it's not working on bronchodilation. You cannot measure it. Well, we were lucky that we participated in a trial with roflumilast at the moment of time where roflumilast got approved for the American market, and that the FDA said, we want a trial to show what the efficacy of roflumilast is when it's given on top of triple therapy. Roflumilast at that time, trajectory was nice, going from company to company, and also spending a huge amount of money to show that it was doing something. So we were the first ones to show what roflumilast was doing on top of triple therapy. And it was interesting. We saw that roflumilast, given orally, was going into the lung through the perfusion side and reaching regions which were less ventilated than perfused. So we saw that the amount of blood vessels that were regionally present in a lobe was indicative for the response to roflumilast. And the response that we looked at was a drop in hyperinflation. Hyperinflation measured regionally based on the lung volume, where we have predictive equations per lobe, what the lobe volume should be based on gender, age, and height. And we could look at regionally what's happening with the hyperinflation. So we saw a drop in hyperinflation, and it was larger where there was more blood vessels, indicating that roflumilast was doing something there. Then the interesting thing began. When you do that, you reduce the hyperinflation, but then you start shifting internal airflow distribution, because if you can exhale better, well, you can inhale better in that region, and more air is going there. And if you add then triple therapy to that, you're shifting that to different zones. And we actually saw that in these patients, where roflumilast was reducing the hyperinflation, you got a more larger deposition of the inhaled triple therapy. And that correlated with changes in airway dimensions, which correlated with change in FEV1. So we generated some data that for the people working on roflumilast was an eye-opener, because in fact adding it on top of triple therapy for them was a good thing. They never dared doing that because they were scared that it would reduce their signal. In fact, it increased their signal, because there's a whole chain of things ongoing that through imaging and looking at different aspects obtained from the imaging, we could make visual, we could quantify it, we could also show it. At the moment, we're finishing a confirmation study to really validate this. Again, validation very important. Next milestone, the interactions with the agencies. We have been in continuous discussion with the agency, mainly the FDA, and last year in June we got a letter of support for the work that we did in IPF, where they stated in the letter of support, for IPF research, FVC was never validated as an endpoint, but it was accepted for the 2 products that were registered to come into the market. And we, as an agency, encourage companies working on IPF to consider this technology to be included in their trials. Very important for us. Next milestone, also a big one. We did a study for GSK, looking at their PI3K inhibitor. The results, where we were primary endpoint in the phase 2A, were used at the JP Morgan conference in San Francisco to explain their status to investors. Biomarker being used to talk to investors, for us very important, because we feel that the market is reacting to that. Investors see this, and investors talk to other companies, and they come to us. Next milestone, which was important, very recently, Galapagos presented the results of their FLORA trial in IPF. Again, we were primary endpoint, co-primary endpoint with FEV1. FEV1 indicated that there was an improvement. We confirmed that. Share price since the communication, again with our technology in it, went up $1.467 billion. Yes, making impact. Next to all these milestones, there's some thoughts that I find important to mention here. Don't be afraid to take a risk. Take a risk. But don't be afraid to walk away from a previous choice. If it turns out that the choice that you made was a bad one, don't insist, walk away. Take another option. Empower and trust your coworkers. You cannot do this alone. You have to work as a team if you want to grow a company, if you want to bring a biomarker into clinical practice. But be ready to intervene. Don't let your coworkers run off in a direction which is different than where you want to go. Good work sells. Very important. But evidence sells better. But selling what the client needs works best. Know the field. At the moment, I'm not afraid to enter discussions with any of the companies in the market, and I'm quite sure that I know the market better than them. Often in big companies, there's so many things ongoing that the people that you speak to are not 100% aware of what's ongoing in the company. So that's the kind of additional value that you can also give to your customers by working with multiple parties. You can guide them, you can prevent them from taking the wrong choices because you've lived it in the past with them, with others. But you can help your client to get to the market in the right way. Well, diversify, generate knowledge, look around. If you can lay your hands on something, try it. It's never bad. At the moment, we're looking into other fields than the respiratory. Our revenue is 100% respiratory. But if we have the possibility to look at something else, we do. There might be something interesting in there for us too. Get your biomarker accepted. That's an important one. Work on the registration. Was mentioned by the previous two speakers. It's important. If you really want to make the difference, in the end, you want to be accepted as a biomarker. And this one is also very important. Support your core activity where needed. When we came with an imaging biomarker into the field, it was difficult because there were almost no CROs offering imaging services. And when they were offering it, was not controlled. They were just taking an image. For us, taking the right image was very important because we wanted inspiration, expiration. And to make sure that this would happen, well, we started providing CRO services. Small, but to facilitate the usage of our biomarker in the clinical trials that we're running within the companies. And don't forget your core. That's something that could also happen when you're supporting your core at some point. Well, the other activities might become more interesting and you might walk away from your core. Stay focused on what you want to do. Some facts about the company. So we exist now 12 years. We grew with a compound annual growth rate somewhere between 30 and 35%. We're still fully privately owned. We never had an external investor. It was all done on revenue. Wasn't easy, but if you're convinced and your data is there to support it, work for it. You can do it. At the moment, we have 35 people worldwide. We have an office in India. We have 2 offices in Belgium. We have an office in Portugal. We have an office in New York, and we're opening an office in Los Angeles. And our presentations nowadays are where they should be. We know what our clients want, and we're adding value into the presentations for our clients. So maybe a small video, or first, no, this one. So we have used it in the respiratory field in all the possibilities that you could run into. Then I have a small video of how it works. So here's the patient. Since the data collection is important, the patient is breathing through a spirometer at the moment of the collection of the data, because if your endpoint is image-based, make sure that the images are correct. So the patients are scanned full expiration and normal, full inspiration and normal expiration. And then you get data from that, the traditional DICOM images, which we segment. And for the segmentation, we use existing software. Again, we didn't want to develop software ourselves. No, we wanted to extract things. And we wanted to extract things linked to the patient, meaning that you need to be able to do this in a repetitive basis. In the beginning, I said one patient, one scan, 2 weeks, 2 scans, 4 weeks. At the moment, we brought it back 1 patient, 1 day to do the whole analysis, which is still a lot, which still needs to go faster. But by reducing the time, we were able to generate more and more useful data for our clients. Also, communicate your results in the right way. You've seen in the beginning, my color scale went from blue to red. For an engineer, that's normal. For clinicians, it's not. When they see blue and red, they don't understand it. For an engineer, red is a lot, blue is a little. No. For a clinician, red is bad and green is good. Details. But it's important details. So right now, our color scales go from red to green. For volume changes, green is an increase. If we would do the same for resistance, green is a decrease, because that's the good thing. Also for the deposition work, you see that our models always become nicer to look at. At some point, we took a deliberate choice. We hired a graphic designer to make our images look nicer. It's still scientific data, but it's just more appealing to look at, which makes it easier for the companies to use it internally to communicate their message, but also to use it externally. This product here, Bevespi from AstraZeneca, their complete marketing program for the US is using our data. For us, this is great, because in the end, clinicians are exposed, and that's where we want to go next. I will touch on that, because our work up until now was in pharma companies, clinical trials, but we want to go beyond that. And if you want to go beyond that, you have to spread the message. In the end, it turns out we don't have one biomarker, we have a set of biomarkers, which also, it brings advantages that you can look at the same patient from different aspects, but it brings disadvantages because clinicians and pharma companies are not used to work with compound endpoints. So this is something that internally we also have invested quite a lot of time in, working on statistical models that don't use one parameter but that use multiple parameters. And especially the difficult part there is come up with sample size calculations that work with different parameters combined. Some facts about our FDA pathway. As you can see, we have 4 pathways which we're currently working on actively. The IPF, we got the letter of support that I mentioned before. We have submitted a full package to the FDA in September. Around COPD exacerbations, we also have a package which is ready almost to be submitted. For bioequivalence, we think we can make a difference there by using simulations in virtual patient databases to bring the time to bring a bioequivalent product to the market, or the feasibility to bring it to the market in the US, to make it feasible. So this package was also submitted, so we have 2 files for which we're waiting for feedback from the agency. This one is going to be submitted quite soon. We're also working on something on the combined presence of COPD, IPF, and PH to see what we can do there. We also have some interesting data in that field. As I mentioned, we want to go further than the clinical trial space. For that, we have been working on what we call BronchoLab, which is actually our gateway to the clinicians. Data can be uploaded and we can provide results back. But for this, we again made a choice which was very much thought through within the company. We didn't want to give the clinicians just parameters. No, we want to go together with the clinicians at this point, set up some IITs to really define what the added value of these markers is in a clinical practice setting, because a clinical practice setting is different than a clinical trial setting. In a clinical trial setting, you want to show the efficacy of a drug. In a clinical practice setting, you want to show added benefit for the individual patient. So which is a new way of approaching the data, and we're working hard on that to get that validation also done. And this is my last slide. So where do we stand today? Well, active for 12 years, participated in over 30 trials, having, well, over 7,000 healthy and diseased patients in our database, working with big pharma, small pharma, device manufacturers, particle manufacturers, academic institutions, CROs. So that's us, and that concludes my presentation.
Thanks so much, Wim, for a fascinating talk, and as I say, broke some of the rules that Liz talked about on the way, but seem to have got there in the end. Any questions for Wim?
That was excellent. So basically you got around the FDA because you let GSK or AstraZeneca sort that out? That was their issue? Because it was innovative and beautiful.
No, but the thing is, having additional data in your packages in phase 2, it's not a problem for the FDA. They want the registered endpoint in the phase 3. But if you generate more data that gives you additional insight in the previous phases, that's up to the company. You have to convince the company that you give them added value in order to de-risk their step to phase 3. And at present, FEV1 or FVC for IPF is still the endpoint in phase 3. But we're generating evidence for them to go with the right population of patients, with the right dose of drug and the right design into that phase 3 to have a highest chance of getting through that phase as easy as possible.
That makes sense. I mean, the other thing I would add to that actually is sometimes you can use a non-validated or non-accepted endpoint to help with, say, an additional label claim. So you'll actually get the drug accepted on something like FEV1, but you might be able to put an additional claim on the label on the basis of additional data from phase 2. So it works in several different ways.
Excellent. It was excellent. Thank you.
Please, continue.
So, back in the days when you were taking 2 weeks to process one data set, I'm slightly puzzled how you managed then to process sufficient data sets in order to be able to data mine all the range of possible biomarkers and look for correlations with endpoints. I would expect you to have needed hundreds of cases to do that, and yet it sounds like you can only do handfuls.
Yes, the first year, yes, but the increase in speed of the processing went up. Well, we went to India, where labor is cheap, so we could have people work on it. So it's trying to find solutions. And the first year, we didn't generate a lot of data. we generated a bit of data, and the first trial that I showed was for us a lot of work to get that processed. I think we spent almost a whole year working on that, but in the end we had data which was pretty convincing to take it further. But then we had to come with solutions to make it faster.
Another question at the back.
I really enjoyed your talk. Just one question, if I may, as a clinician. You have a wonderful spatially targeted biomarker that comes from an image, and you've applied it to diffuse lung disease. Why? And I'm prepared to accept that that's what paid the salaries, but in the long term, why are you not using it for spatially targeted disease processes?
We also use it for that, but we were having the same discussion yesterday at Upwards with the people working on interstitial lung diseases there. Actually, the fact is, for IPF, for example, what happens in IPF is your alveoli are stiffening up. And when your alveoli are stiffening up, you need a higher pressure drop from in the pleura to in the alveolus to stretch your alveoli. At some point, the pressure drop needed to stretch the airways becomes less than the pressure drop needed to stretch the alveoli. So even with the diffuse lung disease, and we have a lot of data to show that, even in a very early phase, you see that the airways are massively increasing in size as compared to a healthy population in a diffuse lung disease, just because of the characteristics of the diffuse lung disease.
Okay. But there are how many IPF patients in the UK?
Not a lot.
About 1,000?
Yeah.
22,000 new lung cancer patients every year.
But I understand the point when you want to go to clinical practice, but the reality in the pharmaceutical sector at the moment is that IPF is very hot.
Sure.
So there's always different choices to take. I think from a clinical point of view, and if you want to go into the clinical practice, well, in the end you want to aim at COPD and asthma because you have a lot of patients there. But you have to get there, and you have to make choices also from a company perspective to be able to do that.
I'd perhaps turn it around slightly the other way from a drug development perspective as well. Is if I have a biomarker in lung cancer that needs me to recruit 200 patients, that's fine, I can do that. I couldn't recruit 200 IPF patients, so actually if I can use something a bit more specific that enables me to get an answer in 12, that's brilliant. So I think you can turn your comment exactly around the opposite way and say, well actually, perhaps it's the smaller diseases from a drug development perspective, but we need the more detailed biomarkers. There was another question.
So I was going to say, so Mahunton, I work at Patware, so I head up the interstitial lung disease group there, but we were very sceptical of Wim's data, but we've looked at it yesterday, and it does sound it does look interesting actually, because the airways with traction bronchiectasis, when you get fibrotic lung, the airways become dilated and are opened up. But I did have a specific question. So all of your work so far has been with pharma. Did you make a conscious choice to do that at the beginning? Did you try and work with hospitals and universities at the very beginning stage?
No, it happened to be within pharma in the beginning. And I have to say that for the first 10 years, we were mainly a research-focused company, and we did some sales. And it's easier to do some sales and get decent budgets for that in pharma than going to clinical centers. So it happened to be like that, and then we didn't invest a lot of effort in the first 10 years to change that around. But we are doing that at the moment.
I think it's fair to say as well, sometimes pharma is quite a good proving ground for these quite difficult technologies, because actually when we're running a very controlled trial, it's actually exactly what you need to test the technology as well before you take it more widely out into the clinical community. So I think sometimes it's a model that works generally reasonably well when it's
And I think if you compare us to other image processing companies, they have gathered a lot of evidence in large cross-sectional trials, which is interesting. but it's very hard to draw conclusions from that on what you would expect as a change to be relevant for a patient. You see a decline or a variation as disease progresses, but it's not always true that when you start treating those patients that trend is going to be in the opposite direction. Sometimes there's other principles that work, and I think the controlled environment of a clinical trial gives you a lot of information there, because you have a very good control on your data. You have little variability from different sources, so it makes it easier to interpret your data.
One more.
Have you tested your CFD technology in blood vessels at all? Because this is very hot in cardiac CT and cardiology, measuring the pressure drop across a narrowing in a blood vessel.
So, short answer, no. A little bit more detailed answer, we're working on that. So at the moment we're trying to correlate some structural measures with the pressures measured invasively. But the challenge there is to convert a complete long tree of vasculature into a CFD model. That's very challenging. But we're in contact since a couple of weeks with a group that should be able to do that. And we're also starting collaborations with them to actually do that.
Anything else? I had one actually.
Okay.
Which is if you could go, but if you could wind the clock back, what would you do differently? Would you do anything differently?
Well, you would always change some details because you have knowledge now that you wouldn't have in the past. So I think there are some choices that we made and that we stepped back from that you wouldn't repeat because you know that it's not going to work. But in the end, I don't know if I would change much, because making those choices gives you insight on things that you don't want to do for that situation, but maybe also things you don't want to do in other situations. So failing is sometimes good.
Okay, thank you. I think on that note, it's time for coffee. So thank you very much to all the speakers in the session. We've kept on time, so we've got reasonably good time for coffee break and any further questions for any of the speakers. So we're back at 20 to 4. Thank you.